All right, I think we're live now. So, hi everybody. This is the Willamette Valley Aquarium Society's first live stream meeting, and we are going to bring you a presenter. His name is Bill Trevarrow. We're actually going to wait just a little bit for some more folks to join and get this party started. Yeah, so I'll do a little, yeah, I see. I'll do a little, a brief, detailed about Bill Trevarrow as soon as we give our friends a few, see what we got going. So hopefully everyone out there is having a great Saturday. I am very sad that we don't have physical fish meetings anymore right now. But soon, hopefully July or August, we will. And that will be awesome. So sorry, this is my first time doing this, so I haven't really figured everything out, and I've never used YouTube, and I've never used all these streaming software that we're using. So my name is Erica Booth. I am the president for the Willamette Valley Aquarium Society, and yeah, so I'm trying my best to make sure that we do something fun.
Shakedown cruise.
That's right. Background. Every once in a while. I kind of had you muted for a little bit, Bill. Sorry about that.
Okay.
But you can talk now. You can talk whenever. So I'm going to do a little introduction here for Bill Trevarrow. He's our speaker this evening. And his shtick is the research uses and the relations of zebrafish to other fish, biologically speaking. And he has ran the Zebrafish Research Facility at U of O, is that correct?
That's correct, yes.
In Eugene, Oregon for several years, and just until about 2006, and then he's bred and sold lots of zebra danios to other research labs for years. And yeah, I think, and you have your PhD in biology, and
yes.
Oh, great! Yes, I actually can see our chat now. We do have a chat going. So what I'm going to do is write down if anyone has any questions while Bill is talking. We can get those answered for you at the end of his talk. And I'll let him do a bit more talking about what this whole thing about zebrafish and their relation to all the fish research that he has done. And so with that said, do ask your questions. I'm going to write them down, and then we're going to get to them at the end. And right now, we're going to switch screens, and you guys will get to meet and see Bill. This is.
Are they going to see me or just my desktop?
No, they're going to see you talking and your desktop as we share this
okay, cool.
meeting. Yeah, so they'll get to see your cool
shirt. Good. Okay. Should I start into it? Okay, so I'm Bill Trevarrow. I've done a lot of stuff with zebrafish in labs over the years, and also sold them or their relatives to labs for research. And I'm going to talk about why zebrafish in particular are used for research so much these days, uh, what other fish they're related to, and why that might be interesting to people. And in doing that, I'll talk about how the relationships are all determined by phylogenetic trees and how those are made. And then I'll walk through a bunch of phylogenetic trees to show where zebrafish are in relation to other fish groups people would be aware of. And also logical history of that area. So I got a lot of slides. Hopefully I'll be able to go through them pretty quick. Okay, so this zebrafish are used for lots of research, most of it involving genetics and development, but also a lot of other stuff. But that's the main two things. And they work together because you can get a mutant in a fish and then it develops wrong. And you want to be able to see what's going wrong when it's not developing properly. And to do that, you got to be able to see the embryos. For that, they produce just tons of eggs, really easy. They're a beginner fish for hobbyists, so they're easy to keep, which means huge manner with machines taking care of most of the stuff. And you can get lots and lots of eggs, hundreds of eggs. And here is a picture of some eggs stuck into a groove in an agar plate, petri plate with some agar in it with a groove in there. And you can pop the eggs in. The eggs are, let's see, pointer here. The eggs are about an eighth of a millimeter across. So they aren't that big. And they develop really fast. You can line them up like this, and you can stick a needle into, this is the egg part, you can stick a hollow needle into there and inject them with all kinds of stuff that people do in labs, like fluorescent dyes to see where the cells go afterwards, or DNA to change the genetics, or RNA to fix up a mutant or make a new mutant, or there's just some put materials where you can look in the embryo, and these eggs and embryos are clear. So you got lots of eggs, they're easy to get. The generations are like 3 or 4 months long, which makes genetics happen a lot faster than a lot of other animals. So these are among the advantages. Let's see. This is a, darn it. No, it ain't working. Oh, here we go. Arrow. This is a movie a friend of mine made of a zebrafish egg developing. Each of these divisions is every 20 minutes. So it's rolling along pretty fast. And then it makes a pack of cells on top of the yolk cell. And then they start moving over. Gastrulation is happening. So new layers of tissues are forming. The head is now at the top. The tail bud is at the bottom. If you look over, where's my pointer? Here we go. Look over here. You got somites developing. These will form like backbones and muscles like between the ribs and stuff. This is the eye. This is brain structures behind it. And this is about 16 or 18 hours after it got fertilized. You can watch all this in a dish under a microscope, which is where this was photographed. And so that makes them really convenient. This is a 24-hour zebrafish, and the tail's grinded, but otherwise it's just kind of bigger than the previous image. Here's the eye, ear, hindbrain, and this is a common stage I worked on in the lab because the nervous system is starting to develop and functioning. It does twitches. If you hit it with a hair, it will respond with a reflexive movement. So it's got a reflex circuit, which would be sensory neurons into the hindbrain, hitting some inner neurons, which then drive motor neurons in the spinal. So you got a little reflex arc there. And so the clarity and ability to see the anatomy is really great in these things. This would be about a 48-hour-old fish. And there's a whole lot of structures you can see here. So this is where the heart is, underneath the, just underneath the head here, heart. This is a sinus full of blood cells on their way to the heart. At this point, this animal has a pumping heart, circulates the blood. Its gills, which would develop here, are not really functioning now. Gas exchange happens as the blood flows over the yolk sac and returns to the heart. And so that's that. There's the eye. There's the lens in the eye. This is the interior of the developing ear vesicle here. That's the notochord, which kind of acts like a bunch of vertebra for the embryo before it develops vertebra. It's a stiff kind of cartilage rod. There's the hindbrain, midbrain, forebrain. Oops. And there's the pharyngeal arches. That's some of these will develop into gills later on by 24 hours or so. These are the somites, which form segmental bones and muscles of the body, mostly like body vertebra bones, ribs, and the muscles between them, and a bunch of other stuff that probably most people wouldn't know about. There's a notochord. Okay. And because they're clear, there's these other cool tricks you can do too. So these guys in Japan took let's see, stop it. Took a fish and embedded it in an auger, so it's just stuck there. Baby fish. I would say probably 2 days old at this point, looking at it. Here's an eye. There's another eye over here. This is the optic tectum. So the eye, light comes into the eye, it activates neurons, and they send processes to the optic tectum where there's kind of a map field of the optic environment of the fish. And you can see activation from this dye, which glows when calcium comes in, when the neurons are active in response to this paramecium, which is swimming around here. So, and they slow it down, but it's always opposite because of the cross projections involved. Let's see. So here's a slow motion. You can see the activation of the neurons in the nervous system as the nervous system is seeing the paramecium, which to me, that's just pretty amazing. But that's a little trick those guys have. So besides zebrafish, there are a bunch of other fish that are kind of used sometimes for research. And mostly it's because they're relatives of zebrafish and they want to make comparisons like zebrafish have this property. It's a little different in this one. Why is that? And also figuring out which ones are more or less closely related. So there are some pretty big zebrafish relatives up here: Figrati, Dangelia, Meghaliensis, and a bunch of Divarios. But there's some small ones: Margaritatus, Erythromicron. I'll show you pictures of these later. And various little tiny fish, Danianellas, and some others a lot of people haven't heard of. There's also some of the tiny ones, the Danianella or the Pedociprus. They are like when a fish is born, it's an egg. Well, and it's fertilized. It's an egg, and then it becomes an embryo. When it hatches out, it's a larvae, and that looks kind of like a tadpole. And then it goes through a metamorphosis similar to a frog metamorphosis, and it changes from a tadpole shape into a mature fish shape, which would be the juvenile of the fish. It's like a mini adult, but it's not really. These pedomorphic species are little fish that from that young stage just kind of freeze but change into adult reproductively. And so that's Daniel is a good example of that. And so by looking at related species, they get to compare different features of their anatomy. Yeah, let's see. Okay, so here's a Danny fish. And here's the brain. You can see right into it. It's got hardly any over the brain, maybe just a sheet of skin. These black things here are individually flat. So they're thin, but spread out so you can see them easily. Here are the gills. And this fish is like not easy to breed. So it's not that great for doing genetics, but it's just awesome for looking at and being able to see things inside of it. Here's an example of a female Danianella, and you can see individual egg cells here, these big round things. And on a few of them, you could see in the original picture anyway, this is a scan of a photograph, so it's not that good, but on some of these, you could even see the nucleus inside the oocyte, inside the adult fish. So that's like kind of a testament to how clear these fish are. You can also see if you focus in on axons moving from the spinal cord down to muscles and stuff like that. There's a lot you can see in these fish. Here is a picture a friend of mine made. He's the main guy I was selling Danio eggs and embryos to. He was doing a comparison of zebrafish, which is a really well-characterized animal for biological studies, with all these other species. Danio meghaliensis, Danio chopri, Danio cari, Albolineatus, Nigrofasciatus, Chiathet. So a lot of these have just stupid common or trade names. Albolineatus, that's Pearl Danio, so that's pretty stable. Cari is Danio or something like that. But a lot of these names, they just kind of like the people selling the fish make them up to sell more fish, so they come up with a new name every once in a while. And I try to avoid those, but you're kind of stuck with those names when you're looking for fish. So there's been a lot of name changes among the Danios since about probably 2000. And there are several reasons for this. First is people started collecting fish in Myanmar, formerly Burma, Vietnam, and Laos, and southern China. And so there's lots of new Danio species. More than 30 as of maybe 5 years ago. There's been lots of recent studies of how these fish are related to each other, because the researchers are working on these fish and they're saying, well, what are the relatives of them that are good to look at? And so then people do studies to figure that out. There are also newer techniques that are better for producing phylogenies, and those have become popular recently. And so a phylogeny is a hypothesis about how a group of fish or whatever you're looking at are related, and better techniques produce better phylogenies, which then people have arguments about which one's right. And those phylogenies determine how the names are made for them. And so the names can change. Traits besides morphology, which has always been around, there's lots of molecular data from single genes or sets of genes, and now just whole genome, just sequence everything. It might be 2 or 4 billion base pairs, and then compare them in a computer to analyze which ones are most closely related to each other. So there's lots of reasons for recent name changes. It's a phase that things go through when there's like competing hypotheses and they're fighting it out. And somebody says, the phylogeny shows this thing should be named this way. And someone else says, well, I think it's this way. It should be named that way. They go back and forth for a few years, and then it settles down into a more stable situation. So the phylogenies are made mostly by doing something like a cladistic analysis, and that involves a cladogram, which is a kind of phylogenetic tree. And so I'm going to go over phylogenetic trees and cladistic analysis so that you can understand where this comes from, hopefully. So the thing about cladograms is ideally you want branches to be from 1 to 2. And when you're talking about something in a cladistic analysis, it's with respect to some location on the cladogram. So in this picture, the level of analysis is this branch point here between C and the DE branch. Things lower down here are primitive traits. They've been there in the predecessors to this population, which founded these guys up here. And changes that happen above this will differ between different branches, and they are derived. So you got the ancestral population, changes are derived, which differentiate these different branches. And then you work through your cladogram doing this kind of analysis at each possible branch point and compare the cladograms, find out which ones fit the data best, and then choose among those as to what cladograms you're interested in talking about. So the ones up at the top, they are living, current taxa. Symbols like this mean it's something that's extinct and they maybe found fossils of it or something like that. These are living taxa, so you can look at their character traits and get data about what they look like. Unless you have a fossil, you infer what the states are back in these ancestral populations that led up to that by comparing and finding what's common in these branches going back to earlier times. So cladograms and evolutionary trees in general can come in different forms and mean the same thing. So all of these are equivalent cladograms. They can be oriented different. Usually it's like this with time going up. So evolution is ascending. Here it goes sideways one way, sideways the other, or it can go down, descending. Where it starts is the root of the tree. The little branches on the end are called twigs. The actual taxa, the species up here, would be leaves. Parts further down are called the trunk of the tree. So there's all this terminology that just comes from tree structure, basically. These are all equivalent. They can be, you can switch things around, but the branch points between the groups are still the same. So you can have something that looks different, but it's the same. BA, AB, the relationships are the same here. Same with this branch could be flipped with this one, but the relationships are the same. Here's also, they can be just drawn differently. They can be drawn with diagonals. They can be drawn with right angle branches, or they can be drawn where time radiates out from the center and then your branches go perpendicular to the radial direction. But this is the equivalent tree of these other ones. It's just radially organized. This lets you put a lot more species into a little space, so people like that. Okay, so cladistics really likes monophyletic groups. And that's because monophyletic groups are considered natural groups reflecting what really happened as the species evolved. And because of that, a good cladogram can be turned into a Venn diagram. And you can look at these groups. And with monophyletic groups, they will be nesting subsets completely inside of another one. So these are monophyletic. A single population here made a bunch of taxa. All of these taxa that came from this population are in this big blue clade. From this point, the ancestral population made the red clade. All of these are in the clade because they're generated from, they evolved from this ancestral population. Similar with the green and the yellow. Problems in the past with taxonomy, some of the people used non-monophyletic groups to organize the animals that they were looking at. So a paraphyletic group here starts from this ancestral population, but doesn't include E here. So that's not a monophyletic group. That's not a natural group in the view of cladistics. And so they don't like those. That would not be a legitimate group to give a taxonomic name. Similarly, this is a paraphyletic group. And the common ancestor of both of these would be way back here. Lots of things are let out, or you could look at it as just combined two things that are not really that closely related. And so I'll show you some examples of those later. Let's see. Similarly, traits are used to define these groups, and the traits can be rated by how useful they are to provide evidence for branching at certain points. And so a shared trait, which might be found in all these species here, would not be informative of a branch point here because it's shared at an earlier point. It's also present in this species here, but you're looking at this branch point. And so it doesn't inform you about whether just these two are related, but not to some other group. And so is beyond the scope of this where you're looking at here. Similarly, it would be good if you were looking at this branch point, but if you had traits that covered all of these, it would not be informative because it'd have information here that doesn't help you make this decision. Ideally, what people want are shared derived traits. And so those are, you start with an ancestral population, something changes right at that point, and this population has a new trait. Maybe it's blue or something, and it's inherited by these species, but not present in these. So it's informative that these guys are at this time. Similarly, if it was you're looking at an earlier branch point, a trait that's found in all of these groups would be an informative trait, but would not be informative in that case, like over here. You can also have an unshared derived trait. So that's if you're looking at considering this branch point, and you found a trait that was only in this one, it's not informative that these guys are all in a group derived from this population here. So those are not informative. The shared derived traits are informative. Fancy word is synapomorphy. Most people don't use that, but anyway, that is the other side of the coin of monophyletic groups. Define shared derived traits. Shared derived traits define monophyletic groups. It's kind of circular. So if you consider how a cladogram might develop over time, you'd start out with an ancestral population. Here's a Venn diagram. After it branches, there'd be a speciation event. This ancestral population has a set of traits, but each of these lineages leading to a new population independently acquires different changes which make them different. So these two sets of traits are different in these two species that have developed from this first group. And then that can go on with further branching as time proceeds, and you end up with your current species, each with a different collection of traits that have been accumulated through their evolutionary pathway, through whichever of these lineages it went. When you're looking at a cladogram, you often want to know what is the most recent common ancestor. If you're looking at these some of these taxa, say A to B, you go back to the branch point that separates them. That's your most recent ancestor. If it was A to E, you'd have to go back to this branch point, which is where they separated. So that's your most recent ancestor, which is usually what people want to identify. And it also is a way to determine which are the most closely related species. So you go back, and the earliest ancestor you come to that's your most closely related species pairs, bringing them together. So these two are more closely related to each other than either of them are to C, because the common ancestor is way back here for C, but up here for D and E. And similarly, this branch point would separate this group from this group. And so it would be more distantly related. And I think that wraps it up for cladograms. Any questions about that? Apparently not. Okay. So if there are any questions, sure. It's a good place to stop. I can hear you. Oh, this is Zoom.
No. Okay. So we do have a question. Thanks. We do have one question from Andy so far. He says, I recently acquired a small group of 8 wild-caught Danio Rio. Do these and any of their offspring that I hope to raise have any scientific value? I'd love to donate them locally.
Wild Danios can be beneficial. I actually started a couple fish lines from wild Danios several years ago, and they were used to find the sex determining locus of the zebrafish, genetic sex determining locus, like a XY chromosome kind of thing. In the lab lines that were generated earlier, like in the 70s or 80s, they had accidentally bred out the sex determining locus. Don't ask me how. But those fish are weird as far as their sex ratios and stuff. And so we got in some wild type fish. So they are useful. But the real use to them is when you can make a breeding line of them and maintain them, because in science you want to be able to go back to the same fish and get more of the same thing and look at it over and over, because repetition is really important for all this. So a handful of fish, probably not that useful. They're different behaviorally than other fish you'd get from a fish farm in Florida. And they have other differences too, but probably they wouldn't be all that useful to a lab. They would just get a line of fish because it's a more dependable source. And then when they publish it, they can say, I use this line X I got from these guys at the stock center, and this is my results. If you want to check my results, you can go get these fish from the stock center and do the experiment yourself. And so that's why your handful of fish probably would not be that useful. But it's a nice idea. And 20 years ago it would have been quite useful, but now there are these lines available.
That makes sense.
Okay.
Yeah.
Any more questions?
Not any so far.
Okay.
Yeah, can continue on.
Okay.
Yeah.
So here's an overall phylogeny of big scale phylogeny of fish. Down here are invertebrates. This is the line that leads to the vertebrates and, among other things, humans. Here is deuterostomes, which is, you can be a deuterostome or not a deuterostome in development, and it's just different ways development happens. This path of development leads to the vertebrates. So zebrafish are way up here, and we'll just walk through some examples of these. So ecdyzoa, that's like insects, arthropods, nematodes, things with a cuticle that they shed and then get bigger in growth and then shed another time and get bigger or change their body shape or whatever. Above them, the Lophotrochozoa are like worms and bryozoans and I think mollusks. Anyway, invertebrates. Echinoderms, starfish, sea urchins, and sea cucumbers. They're invertebrates too, but they are deuterostomes. So they're more closely related to us than insects. And they develop like us early in development. Starting to get towards fishy things, we got hagfish, which don't have a jaw. I believe they have a radula kind of like thing, like snails have for eating. They're pretty primitive. They can make a ton of slime. Lampreys don't have a jaw, have a sucker mouth. They have gills, eyes, and they got a nervous system somewhat similar to vertebrates. Then you start getting up into real kind of more fish-like things, sharks, chondrichthyans. This is Sarcopterygii, which are fish with kind of lobe, fin, limb kind of things, which eventually develop into tetrapods like frogs or birds or people or whatever. So back to normal fish. We got one of my favorites, Cladisca, Polypterus. I really like these guys. They got pretty loby fins too, but they are not part of the Sarcopterygii. Got Chondroistii, which are sturgeons and paddlefish. Gars, Amia, which is a obscure fish that lives in the Midwest. Osteoglossomorphs, which are the butterfly fish and the arowana. And then a whole bunch of kind of more normal fish people are familiar with. Catfish, tetras, knife fish, salmon and trout, pikes, and cichlids, which a lot of people like, are way over here. The zebrafish, however, are over here in the sapriniforms. And so within all these fish, zebrafish, not that closely related to cichlids, pretty close to tetras and catfish and those guys. Now, although this looks little, sapriniforms is actually one of the bigger groups of fish. And it contains lots of subgroups. And the ones we are going to be talking about are in this figure, they're labeled Rasborini. There's fights between these people naming these things. Some people want them Rasborini, some people Danioini. And so there's all these different names available. The Danios are here, and Devarios, which are like giant Danios, are here. But there's lots of other groups of fish here, many of which you wouldn't be too familiar with. Down here is Taniichthyes. That's the white cloud mountain minnow. So kind of looks like a zebrafish or a danio, but it's actually pretty far away. And it's pretty derived out here. So there's lots and lots of these cypriniforms. Right. There's also a lot of danios that don't show up on these diagrams because they haven't been biologically characterized and put onto the cladograms and published as of when I made this slide, which was several years ago. Anyway, so there's a lot of them which you may not have heard of: Aesculapi, Montianensis, Meghaliensis. I don't even know how to say that one. Quagga, which is kind of interesting. Tinwinny. Some of these fish you can get from places like the Wet Spot, which is actually pretty good about bringing in weird, obscure danios. But a lot of them you can't really get. If you were in Europe, they import a lot more of these obscure fish species in Europe. So go to Germany and buy some fish and bring them back. But that may be difficult due to the current rules about bringing fish into the country. So as I said before, lots of names have been changed. I think I should add this slide further up. And there's lots of trade names or common names for a particular species, like Danio dangelia, mustache danio, bigrati, yomo danio, Karai has a bunch of names. Danio hikari has two names, which may be just different species of the same, different sexes of the same species. So I mostly ignore these names because they aren't particular to a species, and people make them up or they'll just like name them incorrectly. It used to really annoy me. Now I just don't care anymore. But yeah, there's lots of them, and you don't hear about them unless you get into really specialist literature. Okay, so moving into the Danionoans, which would be about the Rosbenoran type of group. So the big box would be Danionoani, which is also shown by this bar here extending across many of these branches. And then within that, you got the Danioclade in red. You got the Danionoans, which is the Danios plus basically Divarios. And then you got some other fish down here, which kind of smaller groups in the Danionoani clade. Okay, so this is Danionella, little tiny pedomorphic fish. It's up here on the cladogram. It's not really even officially part of the Danios. It branched off from the Danios earlier than the founding of the Danio clade. So, but it's an interesting fish, and people like it because they can look at things inside of it while it's still alive. Its breeding is unusual for a Danio. Most people think of Danios as being egg-scattering fish that lay non-adhesive eggs. That's typically zebrafish and pearl Danios. It's not actually true for all of them. This fish in particular likes a kind of gravel bed and will dive down into the gravel, lay eggs, and fertilize them in the gravel. And then when they hatch, the little guys swim up out of the gravel. So that's not your normal zebrafish mode of reproduction. Some of the other fish have slightly adhesive eggs that stick a little bit to things, but not a lot, whereas zebrafish are pretty much non-adhesive. This is another group. These are aquarium fish, Isomus, which is up here next to Danianella. So they're the closest relatives to this one. Is this? Their body shape looks kind of similar. And then down here, you got a bunch of what would traditionally be called danios. This is Danio chiathate, the striped version. There are two versions of Danio chiathates, which look quite different. These, every fish looks different because it's got stripes, but they break up into spots, and they do that at different places on different individuals. They got pretty colorful fins. They're easy to breed like a zebrafish. But there's a lot of other fish in here I'll talk about later. These are microrasboras and microdivarios, and this is an example of a paraphyletic group because it's these two groups. The common ancestor would be here. That clade would involve all of these fish, not just these two. These were just thrown together at some point in the past because they're little tiny fish. They don't have a lot of features and they're hard to score to put into a phylogeny. But now there's molecules. Every fish has like billions of base pairs that can be scored for traits to place them at different places in a phylogeny like this. So as a result of that, people then moving stuff around. When they move them around and they have names that don't make sense like this, then they'll come back and change the names of this. This one would probably become a Divario because these are all Divarios down here, except for Inley Cypress. We'll get to that in a minute. Anyway, so that's a paraphyletic group, but these are little tiny fish similar to Danianella. They come from still waters, probably brown under forests, and they're kind of clear and hard to see. But you can see a difference here. This guy has a long, kind of more slender body, similar to the Danianella. Whereas this guy is kind of like, got this little rotund body cavity here. Looks more like a chop rock. This is a group Kila, which is the old name, and I believe it's changed to Laubuca now because of these kind of reorganizations, and then you got to rename them to make sense. So this is you would have microrasboras, Kilas, this might be microdivario or something like that eventually, I don't know. But anyway, this is another kind of fish. It's got big fins and it can jump out of the water well. These are the Divarios. You'd mostly think of those as the giant danios. There is a whole lot of these fish that you never hear about. Inley Cypress here in the middle is a genus from a lake in the Himalayas, I believe. It's nested inside this other group, so that kind of doesn't make sense, but we'll just leave it at that for now. But there's a whole lot of fish that look like giant danios with subtle differences in coloration and stripes. And then there's some others that are completely pretty different. Further down the list here, you got the Sabois genus, which is one species. And it's got iridescence on the side here and then red at the front and back. And it looks really neat. I don't consider it easy to keep though. It needs like soft brown water, I think, to thrive well. This is your domesticated carp, goldfish. There's the carp that it came from. And everybody's familiar with the goldfish, I hope. Let's see. Well, I guess I didn't have an example of barbs there. Barbs are right next to them on this phylogeny. Further down, we got the tench tinka fish. And further down, let's see, I can't even see it with this thing in my screen. Yeah, you have the white-clad mountain minnow. Oh, there it is. Okay. Which is, there are a few species of this, but they look kind of like zebrafish or other danios, but they're pretty far removed from them as far as the phylogeny goes. Their common ancestor would be, oops, I don't know what I did there, would be way back. So not that closely related to zebrafish, even though they kind of look like it. And then among the danios, there are three main groups. There are these Danio, which you might see in stores. They sell juveniles of them that might be about, that big. But I've got some now that are 8 years old, and they're like this big. So they can get like a medium-sized goldfish. And so they're kind of an interesting group. And there's actually, at this, when I made this drawing, they were talking about, the one species, Dangelia. Now they realize that there's like 5 different species living in neighboring areas. They all look kind of similar, but they're different. And so the game of naming these things keeps going on and becoming more complicated. This is a group of chopperai, which has some name I can't remember right now in the trade, but that there are at least 2 other species that look a lot like this with subtle differences. There's Margaritatus, celestial pearl danio was its original trade name, I believe. And then we got Danio erythromicron, and there's another closely related fish to that. And so these they look different color-wise, but the body shape is kind of similar. They're kind of stumpy and thick top to bottom, but not that thick side to side. Okay, then we get to the main group of the danios in which the zebrafish reside. Zebrafish is at the bottom here. Laser pointer here. Above that is a pearl danio, I believe, Albolineatus. This is a chiathet non-spotted version. This is a Meghaliensis. It's actually a much bigger fish than these others. And this is a Nigrofasciatus, which is actually smaller. So, but their body shapes are all kind of same. They're kind of like a side-to-side, slightly flattened torpedo shape. Let's see. Nigrofasciatus, this one, is closely related to the Danio tinwinnie, which is this one. This is the male, that's the female. And you can see how this guy has stripes, but sometimes they break up into spots. If you look at different individuals of this species, there'll be spots here instead of a stripe in some of them. And here it's just gone all the spots. So that's that one. Here we go. This is the Danio chiathet non-spotted version or striped version. This is the Danio chiathet spotted version, which is a fish I like a lot. It really looks nice when it's in breeding condition. Its fins get colored up similar to this, but a bit more red. This next one down is Danio quagga. This is in this group here. I have never seen this in a store, but I know they sell them in Europe. So get on it, wet spot. Anyway, and then this is Meghaliensis, which is a larger fish. And I haven't had this. I don't know how they are to breed, but these guys, the others are all pretty easy to breed, like a normal Danio. Then you got a group related to the Pearl Danio. So Pearl Danio, I think that one. The Danio rosea is a more reddish version of a Pearl Danio, basically. And then down here you got Cari, and depending on who you talk to, there's one or several species of these. They have a stripe, but they also got a lot of this iridescence on the side. So that's this group. And then here you got the actual zebrafish, and this is its closest relative, Danio Aesculapi, I guess, which hardly anybody's ever heard of. But I have bought some of these fish from the wet spot at some time in the past. So those are the Danios. These are the Microrasboras. This would be in the different group from the Danio group here. And we've seen that picture before. Microrasboras. Then you got Keela, I think, yeah, Labuca, I saw that one before. And Microrasbora, which probably is going to get renamed. The body shape looks different from these Microrasboras up here, and it's not closely related to them. Then we got some of the kind of interesting and different Devarios, which don't really look that much like a giant Danio. So these guys are Inle Cyprus. That's a little lake up in the Himalayas somewhere. And I guess it's a big lake, actually. But anyway, their body is not as full in this area as like a giant Danio. And they're kind of longer and more slender. And that seems to hold for this one, Chiensis. And this one, I've never even seen this fish in person, but it looks great in the picture. Metagenesis, I guess. Anyway, this is another kind of striking fish you'll probably never see in a store around here. Chrysotenatus. And then you got Regina, Devario Regina, Apogin, and Caucaensis. Obscure group. Here are the normal giant Danio type Devarios. And there's a whole lot of these. I've got two species that would be in this group, but are not listed in this phylogeny. Devario asamensis and Devario, the giant Burma blood red Danio, which is a Devario. Anyway, they're both like giant Danios, but they got a lot more red in the belly and the base of the tail and some of the ventral fins. They're all really easy to breed if you set them up properly, and they grow really fast, the eggs. Makes them easy to raise. Okay, so that's all the different kinds of fish I was going to show you about Danios. Any questions about that? Okay. Okay. So where are these fish in the world? So the Danios and Devarios mostly are in this heavily cross-hatched area of Pakistan, India, Southeast Asia, and parts of the islands down here. Related groups would be the Barillas, which look a lot like Devarios, live in like Iran and Syria and in this area. Some of those are pretty cool fish. And then there's these others, which I'm not that aware of. Well, Zaco, I can say that, in eastern China. And I don't know if these are aquarium fish or not. I'm just not too aware of them. Some of them are found in Japan. But anyway, that's where they live in the world. When you think of where freshwater fish live, it's going to be in lakes or rivers. And this is a map this guy made of the rivers of South Asia. And so the area we were looking at is something like this. And so you'd expect species to spread most easily within these river basins, drainage basins, and have greater difficulty moving from one to when basin, one place switches to draining into a different river because of a landslide or something, or birds transport eggs on vegetation to a new river and they seed a population there. But this area is interesting to me because Danios are there, and also because the geology is interesting. You got massive rivers coming really close to each other here. You got the Ganges, you got the Irrawaddy from Thailand, you got the Mekong River here, which drains out in Vietnam, in China. And they're all like really close to each other here. It's like really wacky. So how come might that be? And you can look back into geology and get some possible answers to that. You can also consider when, based upon finding fossils of fish, comparing molecular evolution between these different species, you can get approximate times when different clades diverged. So these guys here, Petocyprus diverged from Syndodanio at 3. These spots mean they actually have a fossil there. And that's like 23 or 24,000 million years ago. Danionellas and Isamus diverged 28. So anyway, you can get these from these kind of diagrams. You can figure out approximately at what time these different groups of fish diverged from each other in evolution and how long they have been evolving independently. And that's on scales of millions of years, 18 to 28 here, but some of them more recent, 5 million here. But anyway, then you can look at what was going on with the land at the time when this was happening, because of continental drift is very active in this area. So this guy, Christopher Scotese, has been studying this kind of continental drift stuff all his life. And he's like kind of semi-retired now, I think. But he makes these maps and he keeps making more of them. And so India's here, Southeast Asia's here. This is going backwards in time, 10 million years, 20 million years. 30 million years, 40 million years. So if you go back further, you can see India came from, used to be joined to Australia, Antarctica, and Africa. And then it moved forward and smashed into South Asia. This part here, the light blue part of India, goes underneath Asia and pushes up the Himalayas. I should get my point around again. In this area, and that pushes up the Himalayas. It's moving up, pushing up here while this stuff isn't. So there's this kind of sheer area. That's where all those river basins come so close together, is right in this area where there were probably rivers before in this situation, and they had drainage basins, and they started just mushing together and sliding along each other. until you end up with the situation you get today, which is like this, with all these rivers really close to each other. Anyway, so you can look at where these, oh, here it is. Okay. Pointer. So, like 30 million years ago, India is already smashing into South Asia, and that's when Danios are starting to diverge. And 20 million years ago, it's still in the process. It just keeps doing the same thing on and on as more and more Danio species are created in evolution. So that's kind of the time setting of what's going on there. Oh, anyway, so that's my talk about the fish relationships. And any questions about that? How are we doing for time? Ooh, we're over. Okay.
So yeah, if anyone has any other questions, feel free to please ask now and we can get those answered. We'll have Bill on here for a few more minutes to see if anyone else has anything else they want to say. I actually have a question. My question is,
okay, sure.
just basically, can you talk about how to breed most of the Danios, aside from the one that dives into the gravel and that, just right, for folks to learn?
Well, my basic thing is I have a water system that takes care of my water changes. It's automated, so doing lots of water changes, having good water conditions, that's pretty basic. Most people probably understand that. I've automated it. It's nice in Eugene here. The water's really soft. And so it's just like really good water. And you can make anything by adding salts to it if you want, which is not an issue for these fish. So good water, feed them well. And mostly for when I was like really breeding a lot of these fish, I grew a lot of live foods. I grew moena, which are like small daphnia, which are really well suited to these fish that have little mouths. A big daphnia you'd have to chomp on it a lot to bite it up into pieces. Also, newly hatched brine shrimp, you can feed those to the adults. But also just good quality dry food. And then for the bigger fish, I would use, I used to get California blackworms. The fish love them. I figured out how to keep them in the fridge pretty well. And they would last a couple weeks or more. And they were like egg food for the fish. So feeding the fish well, giving them places where they would want to breed. And there's several ways with zebrafish. Zebrafish are so dumb, you can put two of them in a box, and if you set it up so they can't eat the eggs, they'll just breed in pretty much an empty box, and you can collect eggs afterwards. With some of the other fish, you might have like a tank planted with fake plants on top of a box that has a mesh on top, because you got to keep these fish from eating their own eggs. Most of them will eat their own eggs. but they breed really easily. The Devarios, you can get thousands of eggs from Devarios really easily. Some of these obscure Devario species, I probably could have supplied the entire hobbyist need of the whole nation from my garage by breeding, 2 or 3 of these guys. There's not much demand for them, and they're really prolific. So, let's see, what else about breeding? The other thing about breeding is not so much breeding, but raising the babies. And with zebrafish, you need live food early, basically, is the easiest way to do it. In the old times, in labs, they'd use paramecia, which you can still do. But now people use rotifers, which are a better food. They're easier to grow, but they grow in salt water. So you got to collect them and then strain them out and put them in the fresh water and have the fish eat them before they die. Also, I would use Walter worms, which are like micro worms, but they stay alive in the water for about a week, whereas micro worms would die in a day or 2. So you can just flood a container with micro worms and have food present all the time for these fish. Micro worms or Walter worms, they're really easy to collect huge amounts of if you're set up to do it. And then I would do a lot of brine shrimp fish that were like a week old. They could eat brine shrimp probably even earlier. But then I discovered you can get, there's like expensive, good quality brine shrimp eggs. There are cheap brine shrimp eggs which don't hatch well, but people will decorinate them for you. So they take the shell off, and it's like a little pellet that has all the stuff of a brine shrimp, but it's basically like an egg. And the fish eat those too. And that's even easier than hatching the brine shrimp. And because the eggs are poor quality, they aren't going to hatch. They have to be the cuticle dissolved off. They're cheaper, even though that processing is done to them. They're cheaper than the good quality eggs that you have to do labor to and hatch. So that's kind of my approach to breeding the fish, I guess.
Oh, there we go. So Lisa H asks, question for Bill when he comes to a break. Who made the map of the river drainages in Northeast Asia? Is it available online?
Yeah, it is. This map takes a while for it to switch. Geography. It's this guy who has a company. I think he's in England. He has a thing on Etsy. I bought this for like $10 or something. And he's got them for all the areas of the world. So if there's some area of the world that you're interested in with what the rivers look like, he's got a map of it. And they're just great looking.
That's awesome.
I highly recommend it. His website is down here at the bottom of my figure.
Looks like it says grasshopper geography right down there. Yeah.
Yeah, right.
Perfect.
So if you go there, you can like go to his website, or you can go to Etsy and probably find it searching for grasshopper geography. Pretty cool stuff.
Great. And then Oh, go ahead.
I was going to say, I used to have this hand drawing. I got a map of Asia and I drew in the river basins by hand. It was pretty crappy looking compared to this. This thing's just great.
Andy was saying it's funny how they call Devario anodali the blood red giant Daniel. Okay. I've never seen them with much red aside from the caudal fin.
They can vary quite a lot. And, but yeah, there aren't that many pictures of them. A lot of pictures aren't that great. But also, if the fish get scared when you're taking pictures, they kind of lose their color too. So if you net out a fish and throw him in a little box to take a picture, you know, the fish is probably not too happy. He's probably scared, and he'll kind of be looking faint.
Wow. Yeah. I think that pretty much wraps it up then.
Okay.
I don't think we have any more questions. I was trying to do a split screen here, but I'm not figuring it out. But hopefully for next time we'll get that figured out.
You know, I think it worked pretty well for the first time. Of course, I'm not watching it. I'm giving it. But it's
no, you did great.
Yeah, I'm looking at some of the comments now.
Oh,
Andy
okay,
seems
good.
to know this fish here, so that's nice. Yeah, there's arguments about what species are what and everything.
Mm-hmm. Beckatine says, as a pet species, thinking of Danios available in pet stores, are there any species you think are not well suited in community tanks? What do you think, Bill?
Well, depends what the other species in your tanks are. If you got little tiny fish, you don't want to put giant Danios in there. They'll just eat them.
Right.
Giant Danios are big, fast-moving fish. They live in water that's moving fast, so they can swim fast. And they're kind of fun to watch because of that, but they could end up terrorizing other fish that don't like that kind of thing. But most of them are pretty peaceful. Danios, like zebrafish, smaller fish, they move around a lot. They're always moving, but not like a Trevarrow. And they would be less disruptive of calmer fish, I guess.
Right. I also agree. It's just they're kind of crazy a little bit compared to some other species.
I've had tanks of Trevarrows, which I really enjoy because they, by themselves, they form these groups and they move around real fast. And I'd throw some fish food into a tank, and they would all charge up to the top to get the food, of course. And then they'd do this little kick so they wouldn't end up jumping out of the tank. They'd go up, get the food, and then go back down. And they would often splash me in the face because they're moving so fast.
Right.
And flipping around with their. So they can be kind of rowdy. They can jump out of tanks if they aren't covered, but most of these fish can.
Right. Well, I think that wraps it up, Bill. Thank you so much.
Okay.
And thank you for donating your time to our club to give this talk about zebrafish and why they relate and how they're important to research. And I know I did learn a lot more than I didn't know that I needed to know.
Well, I'm glad you liked it. I enjoy doing this kind of stuff, so it's all good by me.
Yeah, and hopefully we see you again at the club when we have physical club meetings again soon.
Right, yeah, I've been to a few of those.
Right. And just to end this meeting, I just want to say thank you to our members and for all the hard work and the patronage to make this possible. We're using the new club computer to do all of this stuff, and we wouldn't be able to do any of it if it weren't for everyone's support and hard work and dedicated hours making this happen. Also, Central Valley Aquarium says, please ask if he knows about the microfex worms. That is interesting.
What are those?
The microfex worms are I'm trying